OT, and I'm going to be your tour guide throughout the animal cell. And then, once we're done looking at the animal cell, we're going to hop on over to a plant cell and see what the differences between an animal cell and a plant cell are. So let's go. Now, before we start talking about the many organelles in the plant and animal cells, I just want to talk to you a little bit about the different kinds of cells. There's prokaryotic cells and there's eukaryotic cells. Now, prokaryotic cells do not have a nucleus, while eukaryotic cells do. And eukaryotic cells are multicellular, while prokaryotic cells are unicellular. An example of a eukaryotic cell would be the cells found in animals and plants, while a prokaryotic cell would be bacteria. Now let's go see about those organelles. We're going to start off in the nucleus, the brains of the cell. The nucleus is the most prominent organelle in the cell. The nucleus occupies up to 10% of the cell space. Inside the nucleus are genes, or DNA. DNA helps determine the cell's identity. Inside the nucleus is chromatin. Chromatin consists of DNA and protein and condenses to form chromosomes. If you look to the outside of the nucleus, you will see the nuclear envelope. The nuclear envelope is a double membrane which forms the boundary between the nuclear contents and the cytoplasm. Now, if you look back towards the center of the nucleus, you will see the nucleolus. The nucleolus is where ribosomes are synthesized and assembled. As we exit the nucleus, we will see the rough endoplasmic reticulum. The rough ER channels proteins to transport vesicles, attaches carbohydrates to some proteins, and is involved in membrane production through the production of vesicles. Attached to the rough endoplasmic reticulum are ribosomes. These are called bound ribosomes as they are bound to the rough ER. Bound ribosomes are the site of protein synthesis. They produce proteins for use outside of the cell or for the cell membrane. Let's follow protein. Woo! Yeah! Okay. The proteins from the rough ER are packaged into vesicles, which carry the ER products to the Golgi apparatus. The Golgi apparatus is a flattened membranous sac. The Golgi apparatus modifies ER products, produces certain molecules, and produces lysosomes and secretory vesicles. Since we just visited the rough ER, let's see what the smooth ER is all about. As we can see from looking at the smooth ER, it does not have attached ribosomes. That is why it is called smooth, because in comparison to the rough ER, it appears to be smooth. The smooth ER synthesizes lipids, including phospholipids and steroids, metabolizes carbohydrates, detoxifies drugs and poisons, and stores calcium ions. That's a lot for just one organelle. Now that we know all about the smooth ER, let's go look at the mitochondria. Did you know that the mitochondria is the site of cellular respiration and produces ATP from sugars, fats, and other fuel? The mitochondria are known as the powerhouse of the cell because the mitochondria produce energy for the cell to use. The entire region between the nucleus and the cell membrane is cytoplasm, which consists of cytosol. Next up is the vacuole. A vacuole is a membrane-bound sac important in intracellular digestion and the release of cellular waste products. In an animal cell, the vacuoles are generally small compared to the vacuoles in a plant cell. An organelle specific to the animal cell is a lysosome. Lysosomes, like vacuoles, are membrane-bound sacs, but lysosomes are made of hydrolytic enzymes, which are used to digest food, other molecules, and old, worn-out cell parts. Microtubules are fibrous hollow rods that function primarily to help support and shape the cell. Microfilaments give shape to the cell and are involved in the construction of cytoplasm during division in animal cells. Now on our tour we are passing the centrioles, which are unique to animal cells. The centrioles are involved in cell division and are paired structures consisting of microtubules and a 9+0 arrangement. And again, they are only found in animal cells, not in plant cells. Now we're looking at the cell membrane. The membrane has two major functions. It forms a boundary between inside and outside of the cell and controls the passage of materials. Cell membranes consist of two phospholipid layers. The membrane is made up mostly of proteins and lipids. There are phospholipids, fibrous protein, glycoprotein, pore protein, and channel proteins. Also, there's cholesterol in the membrane. The proteins are either peripheral or integral. Peripheral proteins are only on one side of the membrane, while integral proteins go through the membrane. The lipid molecules, which are mostly phospholipids, have a hydrophilic head and two hydrophobic tails. Since we're talking about the cell membrane, I'm going to talk to you all a little bit about active and passive transport. I'm going to contrast the two so you can see the difference between them. The biggest difference between active and passive transport is that active transport requires energy, ATP, and passive transport does not. Another difference is that passive transport is the movement of molecules from a high concentration to a low concentration. And active transport is the movement of molecules from low to high concentration. Active transport transports proteins, ions, large cells, bacterial cells, and complex sugars. Passive transport transports particles that are soluble in lipids, small particles in water. An example of active transport is endocytosis, and an example of passive transport is osmosis. Now we are going to take a quick look at how plant cells differ from animal cells. Chloroplast is an organelle that is unique to plant cells. And chloroplast is where photosynthesis takes place and food is produced using energy from the sun. Another organelle that is unique to the plant cell is the cell wall. The cell wall is just past the cell membrane. The cell membrane provides a protective barrier and maintains the shape of the cell. Now, a plant cell has a vacuole, but unlike the animal cell, which has a few small vacuoles, the plant cell has a central, very large vacuole. And a vacuole in plants is very important because it provides the plant with structure. When a plant is watered well, for example, water collects inside the vacuole, causing the vacuole to provide support for the plant through trigger pressure. And without enough water in the plant, pressure in the vacuole is reduced and the plant will begin to die or wilt. Now that we're done looking at all the cells, I want to tell you all what cell theory is all about. Cell theory is one of the fundamental concepts of biology. There are three basic concepts to cell theory, and they are that cells are the basic units of structure and function in living things, and new cells are produced from existing cells, and then all living things are composed of cells. And that basically wraps up cell theory. Thanks so much for touring the cells with me. Bye!